Hi everyone, we are back live now. I'm Katja. I'm sorry about the delays we had today, but we are now on track. So on this session, we have Paul Rutten, who is founder and CEO of Concert Bio. Paul is exploring the microbial biostimulants area of CEA, and he has a very interesting presentation for us today. Paul, over to you.
Thanks, Katja. So as Katja mentioned, what we do at Concert Bio is we enable growers to harness the potential of the plant microbiome for their crops. Now, as a lot of people here probably know, we human beings have a gut microbiome. We've got a community of microbes inside us that's very important for our physical health, even our mental health. And what is becoming more and more clear over the last couple of years is that plants also have a microbiome that's also very important for their health. And a lot of my PhD was on how plants will recruit the right microbes from the soil around them to help them grow. In fact, if you try to grow a plant in sterile conditions with no microbes, the plant will just die. They've spent millions of years evolving to use and need the microbes around them. The challenge is that a lot of agriculture now doesn't have soil anymore. In soilless agriculture, whether that is in peat or coir or rockwool or something more inert, a huge amount of these beneficial microbes that plants expect to be able to get access to in the soil around them are just missing. And so studies have consistently shown that actually these plants are leaving performance on the table. They could be growing better, they could be more resistant to disease, they could even potentially taste better if we just found a way to put some of those microbes back into these systems where they are inherently missing at the moment. And so what we set out to do at Concert Bio was we really wanted to develop the first microbial biostimulant specifically meant for controlled environment agriculture, specifically meant for plants that are not being grown in soil. And what I mean when I say first is rather than finding another hand-me-down, another bacillus that was really meant for soil agriculture, another trichoderma that was really meant for soil agriculture, we wanted to find microbes that were really meant for plants that are not growing in soil, because that is a unique environment. The plants have unique needs, and a lot of soil microbes really struggle to work or even survive in a hydroponic system. And what we decided to do was, because there was very, very little that was known about how, what microbes are even present in soilless agriculture, what microbes are missing, how do they interact with the plant, it made it very difficult to start to say, okay, what does a beneficial microbe even look like when we don't know what's there right now? So we decided to go with an AI-first approach and really collect as much data as we could about what microbes are there and what microbes are not there in greenhouses and vertical farms, and try to link that with the growth and the yield of these plants. And we were hoping that by collecting enough of that data, the models would be able to start finding connections between what microbes are there or are not there, and how well are the plants performing. And so over the last couple of years, we've collected now 17,000 samples, so from growers all over the world, and we started being able to build these models and find these microbes. And I wanted to just talk you through a little bit what that process looked like. So we started with some very humble beginnings. It was just me in a shared office with one desk. And back then, we actually started receiving samples from growers before I even incorporated the company. You can see some of our first results on the right there. This is some of our first ever samples coming in from growers. What we used to do back then, because we didn't have a lab, is we would receive the samples and then we would send them on to someone else who would process them for us. But one of the things that we realized very quickly was that the existing technology that was out there in the world really struggled to process rockwool and peat and fertigation samples, because no one else had really figured out how do you process, how do you understand the microbes that are in those samples. No one else had ever really done it before. And so we raised some money and we moved into our first lab. Again, still a pretty humble start. All of the company's possessions fit in one car. And that was the lab that we moved into. And what we wanted to do with this lab was really build a space specifically designed to have the equipment and to be able to process samples from hydroponic systems, whether that was rockwool or peat or fertigation. And that's some of our first equipment coming in at the bottom left there, and some of our first scientific instruments arriving there. That lab is now fully operational, and we've been able to process all of these samples in-house for the last 2+ years now. one of the things that we did is the first person that we recruited, actually, the man in orange there is Darren. His expertise was really in processing samples that no one else had processed before. He used to work for the Natural History Museum in London, where he was doing things like, how do you process DNA from a jet turbine fan blade? If a bird has been sucked in, there's not much bird left, and so you have to take a little bit of DNA and try to find out what the bird was. The other thing that we started doing was setting up facilities to be able to run some of our own plant trials. Now, this is probably slightly less professional than some of the systems that some of you are running and used to seeing. We took some catering trolleys, put some pumps on the side of them, we put some lights on the top of them, and that was good enough to start creating a small deep water culture system, but stacked together so we can fit more plants into the same system. So we spent the last almost 3 years now processing a huge amount of samples from 40, 50 growers all over the world, in Europe, the US, the UK, the Caribbean, the Middle East. All of that is now handled in-house. We've also built our first models based on that data. The other thing that we've been able to do with this lab is to start to collect actual microbes from these environments. So we don't just take a sample full of DNA, we actually have these microbes on plates, so that when the computer says, hey, I think this might be an interesting microbe, we actually have that microbe and we can grow it up and test it ourselves. Along the way, what we heard from a lot of growers is that they were very interested in this data as well. They wanted to see what we were learning from the microbiome in their systems. And so we launched a commercial microbiome monitoring service where growers could send us these samples and we could tell them what we were seeing in their samples via a dashboard which we created. And the advantage of the technology that we use, which is DNA metagenomic sequencing, is that it's not like qPCR, where you only test for a small number of things. With DNA sequencing, you test for everything, all of the different bacteria, all of the different oomycetes and fungi all come out of that testing. It's really the good, the bad, and the ugly. And so it's the challenge becomes, how do you interpret that data rather than how do you get enough data? One of the easy low-hanging fruit for us, of course, was what pathogens are present. We can usually go down to the species level because we use third-generation sequencing, the latest sequencing technology that's available. We can tell you what pathogens are there, human pathogens, plant pathogens, and the levels. But one of the things that we learned was that actually just looking at pathogen levels was not a very good way of measuring the health of the ecosystem. And so one of the things that we started doing was to look at more general metrics of the ecosystem, which you can measure with DNA sequencing. So for example, we can measure the balance in the ecosystem. How many beneficial microbes are there versus how many pathogenic microbes, and what's the balance between those? That has often proved to be a much more useful way of measuring health than just saying, is there pythium, is there not pythium in these systems? So we took all of this data, we started building models with it, and we spent several months trying out microbes that the models were suggesting and failing, and even finding microbes that would significantly reduce the yield of the plants. We kept tinkering with the models, we kept training them with more and more data. And then eventually, after nearly 6 months of work, we had our first successful plant trial. That's Rachel on the left there with our very professional catering trolleys. And you can see on the right there that some of those lettuces were much larger than other lettuces. We repeated that experiment several times. We saw really consistently that we had some microbes that seemed to be improving the yield of the plants. And there's 2 microbes in particular that were doing quite well. And so we sent those out to our first commercial grower to do a trial. We grew the microbes up ourselves in our lab. We sent them using very professional packaging in a recycled ice box in a Waitrose bag. And we spent 3 months waiting agonizingly to see what the results would be. There's a picture in the bottom right there of those results finally coming back to us. And they said both of these microbes had worked for them. They saw about a 10% improvement in yield on both of those different microbes. And what was particularly nice is that this grower, Jepco, was actually the first people to take samples, and they were also the first people who got to try this microbe in the real world. Now, fast forward about 12 months, this was all of 2024, we sent these microbes out to as many different people as we take them. We wanted to gather as much data about where they did and didn't work and what their performance was like. And we called the 2 top microbes Overture A and Overture B. And over the course of 2024, we ran about 14 different trials, mostly with commercial growers, mostly greenhouses, some vertical farms as well, also some CROs, so people like the UK Agri-Tech Centre who would do independent trials for us. There was also some universities who volunteered to trial these microbes for us. And the average improvement in yield, and this was all done on lettuce, the average improvement in yield that commercial growers reported to us was about a 9.9%. So we didn't quite hit the 10% that we were seeing in-house. We still got pretty close, so we were very happy with those results. One of the things that we learned along the way was that dosage was very important. So initially we were doing the dosage quite wrong, but now we've really learned what the dosage should be. Even with all that, it's still not magic. You can see that for both Overture A and Overture B, there are growers that they just don't work for. Interestingly, most growers tell us that one of them works much better than the other. Usually, if Overture A works, Overture B doesn't work that well for you. But if Overture B works for you, then Overture A doesn't work very well for us. And we are still learning and still trying to understand why that is. But one of the things that we always recommend to people who want to do trials with us is to try both of them separately. And usually one of them will work for you, but we can't yet predict which one will work for you. We've also had the chance through all of these trials to really understand where does and doesn't this work. So we tested it quite a lot in deep water culture and NFT gutter systems on both peat, coir, and inert substrates. We've seen that that has worked quite well. One of the places that this hasn't worked actually is ebb and flow systems. We've tested that on peat, and the results have been very mixed. We've had some successful trials, but a lot of trials as well that haven't worked. So if you're running an ebb and flow system on peat, then please get in touch. We'd love to work on this some more and try to figure out how we can make it work in those systems. We have also been able to try it on aeroponics, and of course we've done trials now on drip irrigation systems on vine crops, which I'll get onto in a minute. But if you are one of the gray dots on there, if you're running a system and a substrate combination that we haven't tried yet, then please get in touch, and we'd love to fill out this box. I wanted to talk a little bit about how we actually apply this product as well. That's the product on the left there. Most growers are getting it in a jerry can format of about 1 liter. And the way that we get people to apply it is actually to put it into your spray bar, your drip bar, your water tunnel, whatever it is that you're using to put water onto your seeds. And the advantage of that is you have that already, so it's very straightforward for you to do. The advantage for us scientifically is that it's very efficient. You only need a very small amount of microbe to be able to cover these seeds because you're only applying it onto the seeds. You're not dumping it into the fertigation solution. And the other big advantage is that our microbe really benefits from being the first microbe that is getting onto the seeds. It makes it easier for it to compete with other microbes that might be in the system and push them out and establish itself on the plant. And in case anyone is wondering, we have tried this on both pelleted and unpelleted seeds, and it's worked for both of them. That hasn't had an impact on performance so far. So we started selling the product in the UK. There is Jonathan from Jepco, who were the first people to take samples and trial the product. So we were very happy that they were our first official customer. And we now supply them for all of their production every single month. And they've had very good and quite consistent results over the last couple of months, in line with what we were expecting, which I'm very happy to say. A lot of you are probably also wondering, how does this actually work? And this is where it gets a little bit complicated, because the advantage of this AI and this data approach that we've taken is that we don't need to understand what is going on in these systems. The models just find patterns in the data, but of course we as human beings, we want to understand what is going on. The models don't even know what a microbe is, but we really want to understand, how does this microbe work? So I have to be a little bit careful about what I say. There's a lot of information I shouldn't divulge, but one of the things that we've done is we've done a full genome sequence of Overture A and Overture B to see what genes are in there, what is it doing. And one of the things that we found is that Overture A, for example, has several genes that produce plant growth hormones. In soil, it's well known that plants actually expect certain hormones to be produced for them by microbes. And so if they're not getting those hormones, that will reduce their speed of growth. And so it looks like one of the things that Overture is doing is actually producing these growth hormones for these plants, which they expect to get, but ordinarily wouldn't be getting because these microbes are just not there. So to talk a little bit about what's happening next, we're really working on coming to a grower near you. We are getting ready to launch in the US. There's a sneak peek on the left there of what the label will look like in the US. We're going to be calling it Prelude rather than Overture. We've got approval in quite a few states in the US already, including Texas, Georgia, Massachusetts, Minnesota, and a few more on the way as well. We should have most of these done by the end of the year, and we should also have regulatory approval in Europe by the end of the year. So if you're interested in that, then please let me know. We did most of our initial work on lettuce. We started with lettuce because it grows very quickly, so it's convenient for us to gather data, to run trials and move fast. But obviously, lettuce is not the only thing that's being grown in greenhouses. It's not even the main thing that's being grown in greenhouses. So over the last 6 months or so, we started doing trials in other crops, including cucumbers, which we've now done both successful in-house and independent trials in the Netherlands. And what was interesting here was that with lettuce, when you have more yield, you can only really get more lettuce biomass. There's not much else going on with lettuce. But with something like a cucumber plant, you can get a taller plant, you can get a wider plant, you can get more cucumbers, you can get bigger cucumbers. And so it was the first opportunity for us to see where was the energy in the plant going. And what we could see with Overture B, for example, is that some of the energy was going into more cucumbers. It wasn't really possible to get bigger cucumbers because if there's any cucumber growers in the audience, you'll know that cucumbers are harvested at a set size. The supermarket says, I must get cucumbers that are exactly this big. And so you can't really get a bigger cucumber because you always wait until they're that size and then you harvest them. But you can get more cucumbers. And so Overture B was really increasing the number of cucumbers quite a lot. Overture A was actually increasing it even more. But what was interesting is that Overture B, a lot of the energy went into a bigger plant. So it wasn't more cucumbers, but it was a bigger cucumber plant. It was, the height was almost 11% bigger. Now, obviously, we are not professional cucumber growers, but if you are a professional cucumber grower and you have a sense of, okay, I know how I could change and steer this plant so that more of the energy went into the cucumbers and less of it went into a taller plant, then we would love to talk to you about how we can better use this product and then compensate with the way we grow the cucumbers to get more out of it. I'm also really excited to say that we've now done our first independent tomato trials. We didn't have the capacity to do our own tomato trials, so we sent it out to someone else to test it. We had the capacity to try 4 different microbes. We actually, when we got the first harvest result in, they said, one of these microbes has increased the yield by 52.8%. And we thought, wow, amazing, we're done. We're never going to find a better microbe than that for tomatoes. And then in the second harvest, that same microbe actually had decreased the yield by nearly 25%. And so obviously what had happened is this microbe would really push the plant to put a lot of energy into tomatoes. But then at the second harvest, because it didn't have a lot of leaves, the plant had really suffered and was below the control. So it's not always a straight line when it comes to these microbes. Sometimes you want a microbe that is not quite too good because you can actually hurt the plant by pushing it too much into tomato instead of leaf production. You can see that candidate 4 there only improved the yield overall by about 1.7%, whereas candidates 2 and 3, which were much more slow and steady wins the race, those were improving the yield by about 6 to 15%. And so we were very happy with those results. And there's more trials running now. If you'd like to help us try out this microbe and figure out how to best use it, then please get in touch. I want to finish by just thanking some of the people behind the science. We're a pretty small team of 12 people based in the UK. I want to say a special thank you to Lisa, who's our COO, and she's the one who keeps the lights on around here. And also Dr. Stacey Vincent, who is our head of biological R&D. She's the one who's been leading the team that has done all of the science that I've presented here. She's really done an amazing job of finding some of these microbes and showing how well they work. I also want to finish by thanking all of the collaborators. I don't have space to put everyone who's helped us make this possible on this slide here, but a lot of people all over the world, both growers, scientists, and other industry experts have helped us to collect this data and to run these trials. And so I want to say a huge thank you to all of you. We are available for trials in North America and Europe. So if you are interested, please reach out. There's a QR code there that you can scan that will take you to our website where you can submit a form, and then we will be in touch with you very, very quickly. Thank you for your attention.
Brilliant, Paul. Thank you so much. That was really, really interesting. I got to know a little bit more about what you're doing now. Thank you. So we do have a few questions for you from the audience. So one of the questions from Jessica is if you have done any work on how these microbiomes they are actually maintained in the system throughout the cycle. Have you done any follow-up DNA tests?
So we do, yes. We always collect microbiome samples as a matter of course for all of the trials. And the unusual and interesting answer with lettuce actually is that we don't really see our microbes at the end of the growth cycle. The yield still goes up, but by the time we harvest the plants, the microbes seem to have mostly disappeared. So we think what's going on is that they're having an important impact early on in the growth, and that sets up the plant for future growth. But by the time we harvest the plants, they've disappeared. We're still working on what that looks like with tomatoes and cucumbers. Obviously, that is done over a much longer period of time, and we actually keep topping up the microbe there. So we don't know the full answer to that yet.
That's interesting. Thank you. So another question from Joachim is if you have seen any effects in decreasing the pathogenic microbiology.
So with Overture A and B, not so much. We do have other microbes that we're testing out that do attack pathogens. Overture A and B, they help the plant in general, which is still helpful for pathogens, but they don't directly attack a pathogen.
Okay, that's interesting. Also, have you done any experiments with perlite at all?
I think we've done some very small scale trials in like little grow boxes in perlite, but I don't think we've done anything in large scale.
Okay, that's interesting. Thank you. And we have two more questions. So, can it be used on cuttings or in vitro material in the hardening stage?
I think this is a strawberry question. Unfortunately, I'm not a strawberry expert, so I'm not sure. We are running a strawberry trial right now, and the results have been promising so far, but I don't think we know the answer to that question yet, unfortunately.
Okay, so there is one last question. So, what happens to the bacteria in the drained water? Since you are giving them a condition to grow faster than nature, is there a safety issue in the future?
So it's a very good question, and a lot of the samples that we've taken have been in the fertigation solution to see where do the microbes go in the system. And what we've seen is that they only really stay on the roots of the plants. We haven't seen them survive in the fertigation solution. We've even done trials where there were treated plants sitting in the same pond right next to untreated plants, and there was still a very big difference. The microbes don't really survive in the fertigation solution; they survive on the roots, and so they don't really spread through the system. Is the answer?
Okay, thank you. I have also a question for me. So,
Yeah.
I'm working with strawberries. So we have problems often with Pythium and Phytophthora. So
Yeah.
I just wanted to ask whether your approach could be used to find microbes that they could potentially prevent those diseases.
Yes, absolutely. It's actually easier for us to find biocontrol microbes than it is for us to find biostimulants. From the data, it's very easy to build models to do that. The reason we've focused on biostimulants so far has been because the regulation is easier for us to get a product on the market for a biostimulant. But we have microbes that attack Pythium, and we are very excited to put those on the market soon. We're just focusing on a biostimulant for now.
Brilliant. And one last question. I just wanted to ask you because often we have obviously large glass house facilities that they are using UV or other things like peroxide to sanitize or clean anyway than water. Do you see there is a negative effect on your microbes using UV or peroxide?
So we haven't seen an issue in greenhouses. This is again where applying it onto the seeds is very helpful because it lives on the roots of the plant. So if there's a UV filter somewhere in the system, our microbes don't interact with it. It's not an issue. We have had a couple of extreme cases with some vertical farms where there was a lot of sterilization going on,
Yeah.
and I think that did reduce the effectiveness of the microbes. But the sort of things that greenhouses do don't really have an impact. This is a big part of the reason we go for this seed-based application.
Brilliant. Okay. Thank you very much, Paul. I think that's it for now. I'm sure if anyone wants to talk to you further, they can either scan the QR code or send you an email. But also, this platform, Pine, is amazing for you to send emails and communicate with the speakers and between the attendees. But for now, thank you so much, Paul. Very nice to listen to your presentation, and we'll catch up later.
My pleasure. Thanks, Katja.
Bye.
Bye.